Hello everyone. Today we'll be doing our first lecture in biochemistry. So this will be some just basic concepts, basic molecular concepts. A lot of this will be reviewed from our biology series. So if you like more detail on any of the topics, check that series out. So today we're going to be looking at the basic common characteristics of life, what are molecules, and what are the molecules most commonly studied in biochemistry, or at least the ones we will look at. We'll look at the shared molecular characteristics of life, including the law of thermodynamics, ATP as energy, and the small and large biomolecules. So, molecular life. When you break down life forms to their molecular constituents, seemingly distinct organisms actually share the same molecular characteristics. So even here, we can see that all these different things when we get down to the tiniest parts, we're very similar to each other. So again, we'll go through a bunch of basic characteristics of life. So one, homeostasis. So homeostasis is the regulation of the internal environment to maintain a constant state or balance. Examples of homeostasis include the regulation of blood glucose through insulin and glucagon to control our blood sugar, our water balance, so our H2O levels, keeping us hydrated, and our core temperature. So things like our hypothalamus and our regulating our body temperature, and you've seen fevers and stuff like that. Another characteristic is organization. So this means we're composed of one or more cells, which are the basic units of life. So here we go from the whole organism. So for this example, humans. Then we go to the tissue level, which includes our nervous tissues, our muscle tissue, epithelial tissue. Sorry, I spelled wrong. Connective tissues. Then we go down to the cell, which include the neurons, for example. Another characteristic is the process of metabolism. So metabolism is the chemical processes that are necessary for the maintenance of life. So essentially, metabolism, which we've talked about a little bit in our biology lectures, is the balance between catabolism and anabolism. So catabolism is when substances are broken down, providing energy. So we yield energy. Energy is released. In anabolism, substances are synthesized, requiring energy. So energy is put in. Another characteristic is growth. So growth is the maintenance of a higher rate of building up than breaking down. So essentially what we're trying to depict in this figure here is that the scale is leaning towards metabolism. So this occurs when we're younger, and we can see how we grow because there's a higher rate of building up and breaking down. There's usually an increase in size in all parts of the organism and progression from a simpler to a more complex form. And this can occur in our proteins and etc. Another characteristic is adaptation. So adaptation is the ability to respond over a period of time to one's environment. This is determined by genes and the environment. So some examples of adaptation, you can think of your own, but elevation, so altitude. So when we're at altitude, when there's a lower percent oxygen, we can adapt through our red blood cells. We can increase our hemoglobin content to compensate for this. We also have exercise. So when we exercise, we have our exercise adaptations, improving our lung capacity, our VO2 max, different stuff like that, muscle strength, muscle size. And we also have heat adaptation. So this includes our acclimatization to heat. So spending a period of time at a certain temperature, you begin to adapt. Your body begins to regulate at that temperature. So there's many examples you guys can think of, but this course is kind of focused from a kinesiology background. So a lot of our examples will be focused on exercise or sports nutrition. Just keep that in mind. This isn't a true hard biochemistry lecture. Whoops. Sorry about that. Can switch slides. So next we have responsive stimuli, which is our ability to respond, i.e. reaction, in the short term to a stimulus. So action. So again, essentially that the thing is alive. We're able to react to our environments and we're basically able to adapt on the fly. So next is reproduction. So for these cells to continue to live, we must have reproduction, which is the ability to produce new organisms. As we talked in biology, we have mitosis, which occurs in our normal cells, and meiosis in our sex cells. So again, we go from a parent cell, we have DNA replication, and we have two daughter cells, identical to the parent cell. In meiosis, the parent cell, we have DNA replicates, which you have 2 daughter cells, which are different from the parent cell due to recombination, and we can have, then again, these split into further 4 daughter cells. So the other basic characteristic is that all life is carbon containing. So carbon is the chemical element that forms the base of life. Sorry for the messed up symbols in this lecture, guys. Just so I want to point that out right now. Sometimes these question marks will come up. I couldn't fix it. But next, we all have a reliance on water. So, sure you know, H2O is a chemical substance that's needed for survival in all forms of life. So you've probably understood that why there's life on other planets. There's no water, stuff like that. Next, we must have genetic information. We must have our DNA and RNA. And we must be complex. And this means we must be made up of interconnected and related parts. So next question we're going to focus on is, what is a molecule? So simply put, a molecule is the smallest particle of a substance that retains the properties of the substance. So it is a group of atoms held together by chemical bonds. So this isn't our electron level, but this is more of the actual nitrogen or the molecule or water that contains multiple chemicals held together by chemical bonds. Again, it can be composed of one or more atoms. So another easy example is water, which is our H2O, where we also have glucose, which is C6H12O6, and our CO2, or carbon dioxide. While an atom is the smallest particle of an element that retains the chemical properties of the element. So this would be our O2, or our oxygen, the hydrogen, and water. So just the constituents. So this would be breaking this molecule down into its atoms. Oops. So molecules. So biological molecules can contain a few or many thousands of atoms. There are nearly infinite possibilities. So here's serine, which is a single amino acid, compared to alcohol dehydrogenase with enzyme. What you can see is it's just like a huge glob of different proteins held together. So again, we have our structural hierarchy. So the most common molecules you see in biochemistry, or what you see, are carbohydrate, and we'll talk about each of these individually. Our proteins, our nucleic acids, and our lipids, or our fats. But we'll go through each of these next. So even though we talked about these different molecular structures, there are a lot of shared characteristics. So now that we know the common basic characteristics of life, which we mentioned are 10 different characteristics, and what sorts of molecules life forms contain, so we talked about our carbon and our need for water, we can address the question of, what are these shared molecular characteristics between life forms? So all life forms, we have these 3 rules. So life obeys the laws of thermodynamics, which we'll discuss later. ATP is the energy source of life. So we talked about this in biology, how ATP is created in the electron transport chain and through our cellular respiration. And life is composed of small and large biological molecules. So the first law of thermodynamics. So thermodynamics studies the movement of energy, and this applies to many different types of systems, including biochemical. So when thermodynamics, a lot of people think of physics, but this physics in energy relates to pretty much all our bodily processes as well. So typical physical thermodynamic system, in this picture we can see here, the heat moves from the hot boiler to the cold condenser, and work is extracted, in this case, by a series of pistons. So this is kind of how your car engine works in a simpler way. In a biochemical thermodynamic system, energy comes from food, so we break down our food to those energy molecules, and work is extracted by the body. So we take, something's put in, energy comes out. So the first law of thermodynamics is the law of conservation of energy. So this first law states that energy can neither be created nor destroyed. It can change forms. So that means when we intake our food molecule, it doesn't just disappear, this energy is used. So in this example, we intake our food molecules, and with oxygen, so we take that ATP, and with oxygen we form ATP through cellular respiration, we get our chemical energy. And our waste products are carbon dioxide and H2O. So again, if you want a detailed look at these processes, go to our biology lectures. Again, these processes require oxygen because it's the final electron receptor in the electron transport chain. And so we need that to create that ATP in the ATP synthase. And again, carbon dioxide and water are produced as products of food breakdown. The second law of thermodynamics. The second law of thermodynamics is that all processes increase entropy or disorder. So we'll get to entropy and disorder quite a bit in our other lectures, but just keep that in mind. And again, the second law of thermodynamics is energy systems have a tendency to increase their entropy rather than decrease it. So large food molecules have less entropy and are broken down into smaller molecules that have more entropy. So you can think of a big molecule. When we bring it down, it's going to be broken down here. Now there's more disorder per se, and this has an increase in entropy. But again, we'll discuss this in more detail so it makes more sense. Again, this is a process of catabolism. So remember, metabolism, we are breaking down to release energy. Food is broken down and release energy. So one more point here. The opposite is also true. Keep that in mind. So remember, while catabolism is occurring and therefore increasing entropy, there are also small molecules being organized into large molecules and therefore decreasing entropy through anabolism. So we break down that food into our molecules. So we have that increase in entropy. And then now we're going to take these and build my hemoglobin protein so I can transport oxygen. Therefore, we just took another reaction where we decreased entropy through anabolism. So it's a kind of continuous process. Technically, this kind of contradicts the law, but an increase in entropy equals catabolism, but a decrease in entropy equals anabolism. So it's not invalid. It's just a different process. So furthermore, ATP is the energy currency of life, as we talked about. So in biochemistry, there are multiple sources of energy and multiple types of work performed. So again, our energy sources are mainly those macromolecules we talked about, which are carbohydrates, lipids, proteins, and nucleic acids. So these can all be broken down. And the types of work we can do with this energy include muscle contractions, nerve impulses, ion channels such as the ATP pumps, an electron transport chain, heat generation, and pretty much all the processes in our body. Again, something to think about is, does it make sense to have multiple types of sources to produce the same type of that are specialized for each type of work? So again, this is something to think about, and we can talk about it later and how certain things are specialized for certain activities. So ATP, what is it? So ATP is essentially the molecular currency that transports energy within cells for metabolism. So ATP is produced during photosynthesis in plants and cellular respiration in animals and humans. So as we talked about in biology, we get our glucose from eating plants or animals that have eaten plants, and we eat them. That glucose enters our glycolysis. Glycolysis enters the citric acid cycle after pyruvate oxidation, and this goes to the electron transport chain, and then we get our ATP. But again, more detail, go to biology. So ATP is usable for a multitude of cellular processes. So for example, here's a picture of ATP. So here we have our adenosine molecule, which is a nucleoside, which we've talked about in our biology lectures. And we also have a triphosphate, so that's where it gets its name, which is 3 phosphate groups. So again, when we produce energy, we take off the phosphate groups, and now we have inorganic phosphate there. And ATP becomes ADP and energy. But we'll talk more about this later as well. So third molecular characteristic is that our body is composed of small and large biomolecules. So remember, the main biomolecules are carbohydrates, lipids, proteins, and nucleic acids. And each of these can be divided into smaller and large categories, which we'll discuss throughout this lecture and when we take a dive into each of these individually. So, more relevant to our course in this kinesiology focus is we'll talk quite a bit about nutrients and then how they work in our body, and as well as how exercise affects this biochemistry as well. So a nutrient is any substance that is of nutritional value to an organism. Nutrients provide the building blocks for the organism to maintain, grow, and repair itself. So here we have a breakdown of our nutrients. So we have our micronutrients and our macronutrients. So here are those macronutrients we talked about, as well as our macrominerals. And then we also have our microminerals and vitamins. So I'm sure you've heard of many of these. So micronutrients are an essential nutrient that is required in small amounts, like vitamins and minerals. So you guys have also heard of vitamin A, B, C, D, etc. And macronutrients are a nutrient that is required in large amounts. So these include our carbs, proteins, and lipids. These are our main sources of dietary food, and these are the ones we use for pretty much majority of our functions, where these are needed as in much quantities but are still vital. Because obviously you've likely heard of different mineral or vitamin deficiencies can be very serious as well. So here's some examples. Micronutrients. These include all the vitamins, including A, B, C, D, E, and K. Example of some microminerals include iron, copper, magnesium, and zinc. You can look at some food ingredients and you'll see all these different things if you're interested. And our macronutrients, again, are carbs, proteins, and lipids. And our macrominerals include, so again, the macrominerals are just larger and we need more of it. So this includes calcium, potassium, sodium, magnesium, and phosphorus. So again, as I mentioned, nutrition facts. So biomarkers are nutrients, and specifically, they are macronutrients needed by living organisms in large amounts. They are the source of biomolecules in food. So again, here's our proteins, are vital to so many functions. We have our carbohydrates, which is our primary energy source, as well as fat, which is used in energy and also other processes as well. Again, here we see some vitamins as well. So what about nucleic acids? So technically, dietary DNA and RNA provide us with nucleic acids necessary to maintain, grow, and repair our own DNA and RNA. So technically, when we eating that steak or some vegetables, that thing had DNA and RNA when it was alive and still technically does. So we're actually ingesting those nucleic acids as well. And we use these to make our own. And again, we can conclude that nucleic acids are nutrients. They're definitely not reported on labels though. So now we take a look at small biomolecules. So again, we talked about that serine, which is just one amino acid, so it's quite small. So in the simplest form, the biomolecules are found as monomers. Mono equals 1. So a monomer is the smallest possible unit that still retains the characteristics of the molecule. So it's still serine, but it's the smallest version. Small biomolecules fall within arbitrary ranges. So there's several different categories. You have our monomer, a dimer, trimer, and oligomer. So a monomer is 1. You can kind of figure it out. Di, 2, tri, 3. Oligomer means a few, which goes from 4 to 50. And this can work with proteins, carbs, etc. It works for the biomolecules. So our large biomolecules. So in a more complex form, the biomolecules can exist as polymers. Poly meaning many. So polymers are composed of repeating monomers joined by covalent bonds, which we'll talk about later. So polymers contain 100 to thousands of monomers. So again, here's our example of alcohol dehydrogenase, where you can see likely thousands of different monomers. So polymers can be long linear chains, so straight, can be branched, so they can branch off each other, or complex shapes where they're just like something like this. Yeah, so they can be a bunch of different things. So now we're going to take a look into each of the 4 macromolecules, starting off with carbohydrates. So carbohydrates are essentially hydrates of carbon. They contain atoms of the following elements: carbon, hydrogen, and oxygen. This is why carbohydrates are denoted as CHO, carbon, hydrogen, oxygen. So we can have small and large carbohydrates. So our small include our monosaccharides, which include glucose, galactose, fructose, ribose, deoxyribose. So these are our fundamental molecules that see in pretty much tons of structures. Here are disaccharides, which include sucrose, lactose, which you've probably heard about in milk, and maltose. So these are all different kinds of sugars. Then we have our polysaccharides, which include amylose, AMYLOPECTIN, and GLYCOGEN. So now we'll go through each of the monomers. So glucose. So the formula for glucose is C6H12O6. So again, you can follow this formula and look at the structure. So this is alpha glucose, so keep that in mind. So there's a difference between alpha and beta glucose, but So again, we have our carbons. We can see our carbons are labeled 1, 2, 3, 4, 5, and 6. And then we have 12 H's and then 6 oxygens. So glucose is a product of photosynthesis in plants and a reactant of cellular respiration using glycolysis. I'm not sure what's so thick. Glucose is a very common energy source in most organisms, from bacteria to humans. So again, we've probably heard that glucose is the most common energy source in humans. The brain is highly reliant on glucose as an energy source, for example. Glucose tastes sweet, but not overly, and has an LFFFLL designation. So you'll see this when kind of the structure of the molecule, if you look at about a line, but we'll talk about that in another lecture. So galactose. So another monomer is galactose. So again, a molecule, so it's close to glucose, but not the same. We can see here the actual formula is the same, but we can compare the structures. We can see here, as you can see when we switch over, those are switched. So galactose is found in dairy products, sugar beets, and some fibers. Galactose is usually not found in nature, unlike glucose and fructose, but rather combined with glucose in the form of lactose, which is the disaccharide. So galactose individually is usually not found, but it's usually combined with glucose to form lactose. So you'll see glucose. We'll talk about this later, but lactose. These are commonly tested things on your intro biochemistry or biology, so just keep that in mind. Again, this is alpha galactose, and there are variations. So now we have alpha fructose or fructose. So here we can see the formula is similar, again the same, but here we can see some differences. We can see the structure has changed. So look, we can see here we got that structure. Now we have a different type of structure. So fructose is a fruit sugar, which you probably have heard of. It's also found in honey and some root vegetables. Fructose tastes sweeter than glucose. Fructose probably has some bad connotations because you probably heard about it in high fructose corn syrup. But that's kind of a modified version. That's not essentially as normal fructose isn't as bad as that, but we'll talk about that later. Fructose is commonly commercially sweetening agent, so you'll see it added to a lot of stuff to make things essentially just taste better. Overconsumption of fructose has been linked with obesity and diabetes. So just a recap, so we have our small versus large biomolecules. Small include our monomers, dimers, trimers, and oligomers. Large are our polymers, and these have unique quality of polymers. We talked about a unique quality of polymers. We talked about our carbohydrate monomers, which include glucose, both alpha and beta, galactose, alpha, beta, and fructose. So the other ones we talked about were ribose and deoxyribose. So we've talked about these in biology, how these are the foundation sugars for RNA and DNA, which are obviously important, extremely important. So here we see the formula for ribose, which is C5H10O5. We can see here in ribose that the difference is this hydroxyl group instead of the hydrogen at this carbon. Again, ribose is found in RNA and is highly concentrated in brewer's yeast. While deoxyribose, which is C5H10O4, so we can send O5, O4, so that's the difference to remember, is found in DNA. Its natural occurrence is much lower than ribose, and again has the hydrogen instead of the hydroxyl. So now moving on to our dimers. So, lactose. So lactose is C12H22O11. So we can here see we have glucose. We have a beta glucose plus glucose equals lactose. So we have lactose formed by a glycosidic bond between the two molecules. So this is the milk sugar making up 2 to 8% of the solids in milk. There's a bond between glucose and galactose is broken by the lactase enzyme, and a deficiency in the lactase enzyme results in lactose intolerance. So I'm sure you've heard of lactose intolerance. So again, the inability to break this bond doesn't allow lactose to be digested properly. So we need that lactase enzyme, so lactase, to break this bond. And there's actually pills you can get with lactase enzyme that actually you can take before you eat dairy products, and it actually works essentially as to replace your deficiency and break this down. This is essentially formed through a dehydration reaction, which is the removal of water from the 2X hydroxyl group. So now moving on to sucrose. So again, we have a glycosidic bond, but now it's glucose plus beta fructose equals sucrose. So in comparison to lactose. So this formula, again, is C12H22O11. Sucrose essentially is the table sugar, which is also called saccharose. It is very sweet and comparable to fructose. It is the most common food sweetener. So again, these are things I hope you can at least take from this, is to be more aware when you look at your nutritional labels, to say, oh, look, it has added sucrose. Why does it have added sucrose? Is it something that's actually necessary? Is it just giving you more sugar for no reason? Stuff like that. So again, sucrose is an important sugar in plants. It's in pineapple and apricots. So now moving on to our next dimer, we have maltose. So again, maltose is C12H22O11, again formed by a glycosidic bond. So maltose is actually two glucoses. It's alpha glucose and glucose, which forms maltose. So maltose is the beer sugar and malt sugar, and is produced as part of the process of brewing grains. So this is involved in, again, brewing beers and grains and stuff like that. And maltose is the least common disaccharide in nature. So next, we're going to our polymers. So the first polymer we're going to look at is amylose. So amylose consists of repeating chains of glucose monomers only. So remember, glucose monomers only. It can contain several thousand monomers. So again, it's just that glucose, up to thousands of glucose molecules, or monomers. So amylose has only linear regions. There's no branching, so that's important when we look at other molecules. And amylose makes up approximately 30% of plant starch, which gives the storage form of energy in plants. So here we have our amylose molecule. We got those, you see, all our bonds. So this repeats many times, up to 1,000. So we can see our glucose repeating. So next one is AMYLOPECTIN. So AMYLOPECTIN consists of repeating chains of glucose monomers as well. It contains several thousand monomers, so it's quite big. It has both linear and branched regions, and is insoluble. So keep that in mind. Again, it makes up 70% of plant starch. So we had 30% composed of amylose, while the rest of the 70%, or approximately 70%, is AMYLOPECTIN. Next, we have glycogen, which you've probably heard out in there different within our body as a storage source, but we'll get to that later. So glycogen consists of repeating chains of glucose monomers only. It contains several thousand monomers. It has both linear and branched regions at the 1, 4, and 1,6 linkages. And it has 100% of animal starch is contained as glycogen. Again, we have our glycosidic bonds connecting these many repeats of glucose monomers. And we can see that it's both branched and linear. So here's kind of a schematic of glycogen. So here we have our protein core called glycogenin. So here we got our glycogenin. And then here we have those repeating chains of glycogen and tons of branching and linear glucose monomers. And then here, this would just be considered the one glucose monomer or the glucosyl unit. So now moving from carbohydrates, we're looking at proteins. So proteins contain atoms of the following elements. So proteins contain a hydrogen, a carbon, oxygen, nitrogen, and sulfur, which may or may not be present. So again, we have our nomenclature here of our monomers being very small with our amino acids. So this will be approximately 20. There are 20 amino acids, and approximately half are essential amino acids. So that means we need to get it from diet. We can't produce it ourselves. But this also depends on the person and stage of life, which we'll talk about when we get into proteins in their own individual lectures. Then we have our dimers and trimers, so di and tripeptides. And then we have our peptides, which means it's less than 50 amino acids. And then a dimer and trimer would be 2 to 3 amino acids attached together, while an oligomer is an oligopeptide of 4 to 50 amino acids. Then, again, like carbohydrates, we have our polymers, which is greater than 50 amino acids. They can be linear and smaller biological proteins. But actual biological proteins usually have hundreds to thousands of amino acids and form complex globular structures and have folding and are usually in their functional form when this polymer state. So, the basic structure, amino acids. So, again, you've probably seen this here. This is our amino acid structure. We'll talk about the R group soon. So, amino acids are the building blocks of proteins. So there are 20 standard amino acids, or AAs, and they're all with the same general structure and formula. So we can see here they're all with the same general structure. What we'll see that varies is this R structure. Amino acids are formed from the translation of RNA, so which we talked about through the ribosomes. We form our polypeptides, and these form our amino acids. Dietary amino acids are not usually found individually, but are as complexes with other amino acids in the form of proteins. So when we eat a food, we're not necessarily just eating the one serine molecule. It's usually part of a larger complex protein. And we can see here that the R side chain is not an atom. It's different for each amino acid, and it determines which amino acid it is. So essentially, whatever the R group is, that will define whether it's serine, leucine, etc. And it also determines the chemical properties. So proteins, as we'll see, can be classified as acidic, basic, and also nonpolar and polar. And then also essential and non-essential. So we'll talk about our dimers, trimers, and oligomers. So peptide, we can basically distinguish it as the number of amino acids from 2 to 50. So again, a dipeptide is 2, 3 tripeptide, and 4 to 50 is an oligopeptide. There are linear linkage between amino acids, and there's no branching. So this one we have that polypeptide chain. They're joined by peptide bonds, and dietary peptides are not usually found individually, but in the forms of proteins, as I mentioned. So in digestion, we take a complex protein, and then we break it down into the peptide, and then in its amino acids. So we take that large protein, whatever it may be, digest it, and turn it into those individual amino acids, and then we can use it as our building blocks as well. And here we can see we have our peptide bond and our dipeptide. Here we have the formation of a dipeptide. So here we have our amino acids, they're formed together. We have the release of water forming that peptide bond, and now our dipeptide. So polypeptides. Again, polypeptides are amino acids of greater than 50. They involve their linear linkage between amino acids. Again, no branching. They're joined by peptide bonds. And dietary linear polypeptides can be found. And again, in digestion, we go from these proteins to peptides to amino acids. So here's our polypeptide here. So we go from that 2 amino acids to our peptide bond. Then the formation of many peptide bonds forming this large polypeptide, where you can see a bunch of different amino acids, 1, 2, 4, 5, etc. So now in the functional, or at least folded states that we will talk about quite a bit later, is our biological proteins. So biological proteins usually contain hundreds to thousands of amino acids. They can consist of several nonlinear polypeptides, both folded and bonded together, but they're not all peptide bonds. These are complex or globular folded structures that usually needed to be functional. So we need this complex globular folded structure so the protein can actually function properly. Each protein has its own unique amino acid sequence. So lipids. So lipids contain the following elements. They contain hydrogen, carbon, and oxygen. So again, we have our monomers. So we have our monosaccharides, disaccharides. We also have our glycerides and our sterols, but we'll talk about these soon. And you can come back to this and see if you can fill it in. So, fatty acids. So fatty acids, there are many different types of fatty acids found in nature. You've probably heard about saturated fatty acids, unsaturated fatty acids, and trans fatty acids. These are from different sources of food, and they confer different chemical and health properties. Fatty acids are often found as part of triglycerides, which are also sometimes referred to as triacylglycerols in food and the body. So the structure of this fatty acid is the CH group, where we have our hydrocarbon and also our carboxylic acid. So here we got our carboxylic acid, and then here we got the long hydrocarbon. So glycerides, so monoglyceride. So aka monoglycerol, like I mentioned, there's also triacylglycerol. So this contains one fatty acid and a glycerol. So the biochemical importance of monoglycerides is that it acts as a messenger in cell signaling, for example, and like diglycerides, a common food additive It is a common food additive used to ensure blending between oil and water. So you see this between stuff like peanut butter, where if you have natural peanut butter, you know there's that oil, but they can add something like this to keep the oil together and stuff like that. So now the diglyceride, so also known as diacylglycerol, this contains one fatty acid and one monoglyceride. The biochemical components, again, it can act as a messenger in cell signaling and is also a common food additive to ensure blending between oil and water. And here's our general structure of our diglyceride. Now we have our triglyceride or triacylglycerol, which is one fatty acid plus a diacylglycerol or diglyceride. It is the dominant form of lipid, so you'll probably see this most referred to in the literature and just work in general. It is the most concentrated form of energy storage, so it's very highly energetic when it's broken down. It can provide lots of energy. The fatty acid makeup of triglycerides is variable, and high triglycerides are a risk factor for cardiovascular disease, which you've likely heard of. So another examples are sterol or cholesterols. So cholesterol or cholesterol is considered a sterol, which is a combination of a steroid and alcohol. So here in this green square, we can see the steroid part, which is 4 fused rings. So 1, 2, 3, 4. And then here we have higher alcohol part, which is the OH on the bottom left. The biochemical importance of cholesterol is quite important. They're a key part in our lipid. They're a key lipid in all our cell membranes and help regulate membrane fluidity. And most cholesterol is made up in the body, and the rest is derived from the diet. Again, you've probably heard of high cholesterol being a risk factor for cardiovascular disease, atherosclerosis, different stuff like that. So 4, nucleic acids, which again we mentioned as, yes, they are a nutrient. So nucleic acids contain atoms of the following elements: hydrogen, carbon, oxygen, nitrogen, and phosphorus. So our monomers would include our nucleic acids or the nucleotides, which is the sugar, base, and 1 to 3 phosphate groups. And then the large form would be the DNA and RNA, which contain longer chains of fatty acids and which hold our genetic information, which we detailed a lot in biology if you want to take a look. So, recap. Carbohydrate polymers include our amylose, AMYLOPECTIN, and GLYCOGEN. Our protein monomer or amino acids forming peptide bonds. This results in yielding longer peptides. Then we have our folding, resulting in our biological proteins. Our lipid monomers are our fatty acids forming the ester bonds, yielding our monoglycerides, diglycerides, and triglycerides, which again is the most common form. And we also have our nucleic acid monomers, which are nucleotides. So, nucleotides. So again, we talked about these bases quite a bit in biology, but again, we have our ribose, which is OH, and our deoxyribose is H, and then we have our different groups, our adenine A, guanine G, which are purines. Then we have our pyrimidines, which are cysteine, uracil, and thymine. Again, uracil is used in RNA. So again, we talked about our base pairing, but again, these have 2 hydrogen bonds, these have 3 hydrogen bonds, but we'll talk about it later. So again, DNA. DNA contains the genetic instructions that underlie the development and function of all living organisms. It's organized into chromosomes, as we've talked about within cells. There is base pairing and is double-stranded and has the deoxyribose sugars. When you consume food, such as a plant or animal, you consume their DNA, which is broken down during digestion into nucleotides that can be then used by the organism. So we essentially get back those nucleotides and use them in our own DNA structure. Sorry, that's supposed to be an arrow. Bad arrow. So as we talked about, we have our base pairing with A to T and C to G. So again, C to G contains 3 hydrogen bonds. A to T is 2 hydrogen bonds. And A to T is in DNA, and then where in RNA it would be A to U. So RNA. RNA is the messenger that carries the instructions from DNA from the nucleus of the cell into the cytoplasm. Complementary DNA is a single-stranded. It contains ribose sugars and uracil, as we mentioned. Here's our uracil. As with DNA, when you consume a food, you consume their RNA, which is broken down during digestion into nucleotides that can then be used by the organism. So again, that's our basic introduction into the kind of the 4 macromolecules that we'll be discussing quite a bit. And as we go through this series, we'll talk about all the different processes of how they're actually used and broken down. So this is just that general structure of the things we're going to be talking about in much more detail. So I hope you enjoyed it. I hope you guys see you in the next lectures. See you next time.